Hi, my name is Amelia Cervera. I work at IBMCP in Valencia, Spain, and I'm going to talk about some novel hepatitis delta-like agents that contain hammerhead ribozymes that we have found in a number of animal metatranscriptome samples. First of all, I'm going to introduce the family of small self-cleaving ribozymes, which is composed of 9 members. The first 4 were discovered in the 80s and 90s. The hammerhead, hairpin, hepatitis, delta virus, and satellite ribozymes were discovered in different infectious agents like viruses and virus satellite RNAs. And the remaining 5 members, the GLMS, twistor sister, and hatchet and pixel ribozymes, were discovered later on in the 2000s by bioinformatic methods. Despite their very different structures and active sites, all small self-cleaving ribozymes catalyze a transesterification reaction in a sequence-specific manner in which a 2' oxygen group attacks a 3' phosphate group, and this produces 2', 3' cyclic phosphate and 5' hydroxyl ends. One of the first members of this group of ribozymes to be discovered was the hammerhead ribozyme. This ribozyme is composed of 3 helices, 1, 2, and 3, that surround that conserved core of 15 nucleotides. And the cleavage point is located between helix 1 and 3 at the point indicated by the arrow. The hammerhead ribosome was given this name because its 2-dimensional structure resembles a hammerhead shark, although in reality the structure is more like the letters Y or gamma because of the interactions between loop 1 and 2, which are required for activity in vivo. The hammerhead ribosome was first described in subviral agents such as viroids and plant viral satellites, which have in common that they are all circular RNAs. And in all these genomes, they play a role in the rolling circle replication mechanism, producing unit-length copies from multimeric RNAs. However, since 2010, we know that they are widespread throughout the tree of life and are found both in prokaryotes and eukaryotes, including humans, and also in phages. There are 3 topologies in natural hammerhead ribozymes, depending on which helix is open. So when the first helix is open, we have type 1 hammerhead ribozymes. When the second one is open, we have type 2 hammerhead ribozymes. And when it's the third one that is open, we have type 3 hammerhead ribozymes. All types are found in prokaryotes, but besides that, type 1 is found mainly in animals, whereas type 3 is found in plants and in viroidal RNAs. Another member of the family of small self-cleaving ribozymes is the hepatitis delta virus ribozyme. This ribozyme has a nested double pseudoknot structure with 5 helices, P1 to P5, and it promotes its self-cleavage at the nucleotide just 5' to the ribozyme sequence. The hepatitis delta virus ribozyme was initially described in the hepatitis delta agent, or hepatitis D virus, which is the sole representative of the delta virus genus. This is a human-specific virus that has a circular RNA genome of about 1,700 nucleotides and contains ribozyme motifs in both polarities. In a similar way to what happens in viroidal agents, the ribozymes are essential for the replication of the virus through a rolling circle mechanism. Besides the ribozymes motifs, this virus contains an open reading frame which codes for 2 different proteins, the small and large hepatitis delta virus antigen. The hepatitis delta virus depends on another virus, the hepatitis v.1, for assembly of viral particles released from the host and entry into new cells. And when a co-infection with both viruses happens, the hepatitis symptoms are much more severe. Besides within the hepatitis delta agent, the hepatitis delta virus ribozyme motif has been found in the genomes of different organisms, and in many cases these motifs are embedded within transposable elements such as line retrotransposons. Hammerhead ribozymes, on the other hand, have been associated with transposons such as Penelope-like elements and retrozymes. Interestingly, both plants and animal retrozymes produce circular RNAs with hammerhead ribozymes as retrotransposition intermediates. Until recently, the hepatitis delta virus had only been detected in humans. However, since 2018, different hepatitis delta virus-like agents have been identified in metatranscriptomic samples from different organisms, for instance in ducks, in snakes, in newts, toads, fish and termites, and also in rodents, bats, deer, hedgehog, and in another bird. The newly described hepatitis delta-like agents share many characteristics with their human counterparts. They all have circular RNA genomes of a similar size with a rod-like structure and code for antigen-like proteins. Most of them also contain hepatitis delta virus-like ribozymes, just downstream from the open reading frame in both polarities. However, in one of the papers, no ribozymes were described for these viruses, and it was even suggested that they were not present. And this is quite surprising, since the ribozymes are required for the virus replication. So we set to analyze if the putative delta viruses from newt, fish, toad and termite didn't really have any ribozymes motifs. And we used different bioinformatic approaches like homology searches, sequence alignments and secondary structure searches to analyze the 400 base pairs that are just downstream from the open reading frame. And with these approaches, we found indeed hepatitis delta virus ribozymes in both polarities of the newt and fish viruses. These ribozymes have a structure that's slightly different from the human one, but they are still recognizable. In the case of the toad and the termite viruses, however, we couldn't find any hepatitis delta virus ribozyme motif. Surprisingly, what we found in the toad and termite sequences were hammerhead ribozymes, and in both polarities. These ribozymes are type 3, which is the type found in viroidal sequences and also in plants, but they have an unusual structure that we have called extended type 3 hammerhead ribozyme. These ribozymes have longer than usual helix 1 and helix 2, and the interaction of the terminal loop 1 takes place with an internal loop in helix 2, which is absent from the canonical ribozyme. Besides that, these ribozymes also show an atypical cleavage site, which is predicted to not work as well as the canonical one. So going back to this previous slide, we can say that yes, all hepatitis delta virus-like genomes known to date contain ribozymes, but not all of them are hepatitis delta virus ribozymes. Some of these putative delta viruses contain hammerhead ribozymes instead. After identifying the missing ribozymes, we wanted to know if both the hepatitis delta virus ribozyme and the hammerhead ribozymes were active. And for this purpose, we cloned ribozymes under the control of either the T7 or the T3 RNA pol promoter and carried out run-off transcriptions for an hour. And after that time, we separated the RNA cleavage products by page, and we saw that the new ribozymes were in fact active with a self-cleavage percentage of about 40%. And also were active the fish virus hepatitis delta virus ribozyme, although the final percentage of self-cleavage was somehow lower. In the case of the hammerhead motifs, they also proved to be active, although the one in the toad virus was much less efficient than the one from the termite. But this was in agreement with the differences seen in the self-cleavage site sequences. So to sum up, we can say that all hepatitis delta virus-like genomes known to date contain ribozymes, but that these ribozymes are not only the typical ones found in the human hepatitis delta virus, there are also hammerhead ribozymes, and these hammerhead ribozymes motifs have an unextended type 3 architecture. It has been proposed, in fact, that these hepatitis delta virus-like agents with hammerhead ribozymes are called epsilon viruses rather than delta viruses. And there are still some questions that need to be answered, like we still don't know how delta viruses evolved, and we don't know what's the relationship between the hepatitis delta virus ribozymes and the hammerhead ribozymes in these agents, and also if there could be other self-cleaving ribozymes in hepatitis delta virus-like RNAs. Finally, I'd just like to point you towards another talk at Mbabayon's talk on June the 2nd, where he'll explain how they found, using the platform Ceratus, lots of additional delta viruses and epsilon viruses in environmental metagenomic samples. And they have even found two new classes of viruses that they have called eta and zeta, which are much smaller than delta viruses, and that they don't have the usual hepatitis delta virus like antigens, although they still contain ribozymes. So that's all. And this is us at Marcos de la Peña Lab in Valencia. Raquel Ceprián, Maria Pedrote, y Mar Bañús also contributed to this work, and also John L. Casey from Georgetown University. And just in case you're interested, Marcos is also presenting a poster in session 4 about insect circRNAs with hammerhead ribozymes. And thank you for your attention.